So let's do a revision of third week of embryonic development, and here you will know what are important events taking place in the third week of embryonic development. So the first one is about the formation of notochord. So here these are the steps. So here see that this is a diagram. So this is the bilaminar germ disc which contains epiblast, hypoblast, and this is the amniotic cavity which is completely lined within the epiblastic cells, and this is the yolk sac which is completely lined within the hypoblastic cells. Now here you see that all the hypoblastic cells, they are cuboidal in shape, but here at a point where these hypoblastic cells, they become columnar, from cuboidal to columnar, and that leads to the formation of prochordal plate, and this prochordal plate is formed towards the cranial end. So this prochordal plate, so here marks the here cranial and caudal end of the embryonic disc. Now second, you see that this is the initial diagram. So here initially not a single epiplastic cell was proliferating into the amniotic cavity, but now see that the epiblastic cells, they are proliferating into the amniotic cavity, and due to the proliferation of these epiblastic cells in the amniotic cavity, primitive streak is formed, and this primitive streak, if you see that, it is formed towards the caudal end, because that this prochordal plate is the cranial end. So this is the cranial side, this is the caudal side. So therefore that I written, so this is the primitive streak, so this is the cranial end, so this is the cranial end, and this is the caudal end. So here important thing is the formation of primitive streak is done. Now here see that, so in the primitive streak, this is the cranial end, and the cranial end of this primitive streak, so here becomes thickened and it enlarges to form an elevated structure, and that elevated structure is known as primitive node. Now you see that the center of the primitive node, at the center there is a cavity formed, and that cavity is known as primitive pit. See, so therefore, so first the formation of primitive streak is done, then the formation of primitive node is done, then the formation of primitive pit is done, and this primitive pit formed at the center of the primitive node. So this is the diagram in this, if you see that. Now see that that these are the proliferative, so here these are the proliferative so here epiblastic cells which are present in the primitive streak. Now see that these proliferative epiblastic cells, they are migrating cranially and also in the midline, and also in the interval space between the epiblast and hypoblast. Now due to the cranial migration of the proliferative epiblastic cells in the midline between the epiblast and hypoblast, and this leads to the formation of notochordal process. Now yes, in the midline. Now here see that, and in the notochordal process, so here that this primitive pit is extending into the notochordal process or the cavity, and due to the result of the extending of that primitive pit in the notochordal process, notochordal canal is formed. Now here see that this is a notochordal canal, and here this is the epiblast and this is the hypoblast, so it is not connected with the hypoblastic cells. Now here see that this floor forming the cells of the notochordal canal, so here mixed with the hypoblastic cells, and this is known as intercalation in embryonic hypoblast or embryonic endoderm. Now here why this is embryonic endoderm, because in future that these hypoblastic cells will form endoderm. Now here see that in the floor forming cells of the notochordal canal, so here cavities are made. Now here you see that due to that cavity, now here that this notochordal canal is connected with the yolk sac, and that notochordal canal is also connected with the amniotic cavity through this primitive pit. So now you can say that this notochordal canal is connected with the amniotic cavity through the primitive pit. So this is the primitive pit, and due to the cavities so here formed in the floor forming cells of the notochordal canal, it is also connected with the yolk sac. Now due to this also you can see here basically nutrition can be transferred from amniotic cavity into the yolk sac also. Now see that initially all the cells of the notochordal canal they are curved in shape, but now you see that these notochordal canal cells they have been converted to flattened cells. As a result notochordal plate is formed, but this is reverse. So soon again that this notochordal canal cells again become curved. Now when the notochordal plate converted into a curve-like structure from a flattened structure, and then the a definitive notochord formation takes place. So that's all about the formation of notochord. So therefore, if you remember all the points through the diagram, then this becomes very easy for you. Now here one thing that a notochord formation occurs when the proliferative epiplastic cells migrate or pass cranially in the midline between the epiplastic cell layer and the hypoplastic cell layer. Now here also that is for the formation of intraembryonic mesoderm also, that is proliferative epiplastic cells migrate in the midline space between epiplastic cell layer and hypoblast, but they don't migrate in the midline. So friends, let's discuss about the granulation process. So now again, so this is the epiplastic cells and these are the hypoplastic cells, and there is a primitive streak containing proliferative epiplastic cells. Now here again this is the interval space between epiblast and hypoblast. Now here see that this proliferative epiplastic cells they are again migrating in the interval space between the epiblast and hypoblast. As a result that so you can see the intraembryonic mesoderm form and the ectoderm. So here epiplastic cells converted to ectoderm and the hypoplastic cells converted to endoderm. Now there is a very important question is that now when these proliferative epiplastic cells now now friends, so this is a diagram so you are given in your book IBC. So see that when these proliferative cells migrate in the interval space cranially in the midline between the prochordal plate and the primitive node, then only notochordal process is made in the midline. But when they don't migrate midline but they migrate anywhere in the interval space between a primitive node and the prochordal plate, then intraembryonic mesoderm forms. So therefore that is notochordal process or the notochord formation and the formation of intraembryonic mesoderm done simultaneously. So therefore that is one very important part that I have written that. So here simultaneously along with the formation of notochord, the proliferative epiplastic cells migrate in the interval space. So between the epiblast and hypoblast to form intraembryonic mesoderm, and this epiblast and hypoblast converted to ectoderm and endoderm. Formation of epiblast, intraembryonic mesoderm and hypoblast is a process known as gastrulation. So friends see that when these proliferative epiplastic cells migrate cranially because towards the prochordal plate in the midline between the primitive streak and her prochordal plate, then only see that notochord formation occurs. But simultaneously that some of the epiplastic proliferative epiplastic cells migrate in the interval space also between the epiblast and hypoblast, and this leads to the formation of intraembryonic mesoderm, and therefore the formation of both notochord and intraembryonic mesoderm so takes place simultaneously, and that point that I want to cover here. See that? So here simultaneously along with the formation of notochord, the proliferative epiblastic cells migrate in the interval space between epiblast and hypoblast to form intraembryonic mesoderm. Now epiblast and hypoblast converted to ectoderm and endoderm, and that formation of ectoderm, intraembryonic mesoderm and endoderm is known as gastrulation. So see friend, this is the correct formation of ectoderm, intraembryonic mesoderm and endoderm is known as gastrulation. So now let's the formation of a neural tube. This is very important. So as you know that this gastrulation process and notochord formation, as you know, so here they take place simultaneously. Now see that this is the ectoderm and this is the portion of ectoderm lying over on notochord. Now see that the portion of ectoderm lying over on notochord becomes thickened to form a structure, thickened plate-like structure. This is known as neural plate. Now see that this neural plate has been buried, and due to burying of this neural plate, that the central part is known as neural groove, and the margins of the neural plate have been raised, and this raised elevated margins known as neural folds. Now here see that this is again surface ectoderm. So here that these are a surface ectodermal cells which are present between the surface ectoderm and the neural fold. Now see that, so here approximation of the neural folds happening, and you can see that as a result of the approximation of the neural folds, that is neural tube has been formed, and also the two surface ectodermal cells that differentiate into also neural crest cells. Now though the neural tube has been formed, it has anterior and posterior neurophores, and due to this anterior and posterior neurophores, it is not completely formed, and also due to anterior neurophore, it is connected to amniotic cavity to get nutrition till the complete circulatory system develops. And finally, when the both the neuropores of the neural tube is closed, then the completely neural tube is formed. That's all. And also the deformity or that failure of the closure of anterior neuropore leads to anencephaly, and failure of closure of posterior neuropore leads to spina bifida. Now as you already know, that is an intraembryonic mesoderm form, and on the basis of the location of notochord, that intraembryonic mesoderm divided into three parts: paraxial mesoderm, intermediate mesoderm and also lateral plate mesoderm. That's all. And also and you know that the formation of the chorionic villi, primary, secondary and tertiary chorionic villi, and in third week of embryonic development also the continuation of the chorionic villi occurs, so the primary villi converted into secondary and tertiary villi. When I taught you the revision of the second week of embryonic development, that is I stopped in the primary villus part, that is in second intraembryonic life. In second week of intraembryonic life, only the formation of primary villus is completed and the formation of secondary villus occurs, and the remaining, basically the formation of tertiary and chorionic villi completed in the third week of embryonic development. So this is the, so here tertiary villi. So see that this tertiary villi, so here consists of, so here basically that extension of the intraembryonic mesoderm, extension of cytotrophoblast and also blood vessels. This is also a very important part, that in third week of embryonic development the formation of tertiary chorionic villi completed. That is also important. So therefore again once again I am going to discuss what are important events of third week of embryonic development. The first very important is the formation of notochord, and the second one is the gastrulation process, where the formation of intraembryonic mesoderm turn and the epiblast converted to ectoderm, and the hypoblast converted to endoderm, and the formation of neural tube, and the complete formation of tertiary colonic villi turn, and also subdivisions of intraembryonic mesoderm. All these are the important events taking place in a third week of embryonic development. That's all.